Welcome everybody to this edition of the ISCB Academy series, a webinar on computational and data resources for COVID-19 and our current pandemic. This seminar is entitled Early Publication Access and EMBL-EBI Biomolecular Data Tackle COVID-19. And we have two speakers, Michael Parkin and Matt Pearce, who are both from EBI, and I will introduce them in a minute. Before I do so, I want to just give you the context of this webinar. This webinar is actually not standalone, but it is a part of a series of webinars on data sharing and data access and data repositories. As ISCB, we realize that data are our main stake. Without data, we're all comatose. And so how you access data and how you can share data is really the lifeline of our research. And realizing that, we have decided that we have a series of webinars on this topic. This is edition number 2. Edition number 1 was on the Protein Data Bank on the occasion of its 50th anniversary. And we had a nice webinar of Helen Berman, who not only talked about the database, but also about what a challenge data sharing was and coming up with all these concepts 50 years ago and as this development unfolded. And so this is our second edition dedicated to the data sharing issue with COVID-19. It is basically, I see it also partly in preparation of a feature that we will have in the upcoming ISCB 2021 conference, our flagship conference of our society, which will happen at the end of July, in the last week of July. And on July 30th, we will have a panel on data sharing regarding viral sequence data before the background of what you may also have perceived as a partial controversy of how data should be shared in such a setting, a pandemic setting where there is a large urge of adequate data sharing. And this panel will not be the last edition. We will continue this series. So after these motivating statements, I'm very happy to introduce the two speakers. And I will start with, I will start with Matt Pearce, who is going to be our first speaker. Matt Pearce is the technical project lead on EBI Search, which is part of the web production team at the EMBL-EBI, the European Bioinformatics Institute. His team provides search services across EMBL-EBI and unified search interface across many of the EMBL-EBI datasets. Matt's background is in software development, having specialized in search over the last 10 years or so. He joined EMBL-EBI 6 months ago. Previously, he was involved in the BioSol-R project, working to improve search for bioinformaticians. Our second speaker, Michael Parkin, is a data scientist at EMBL-EBI. His work involves sourcing new content in Europe PMC, which is a literature database, ensuring data quality and analyzing trends in the literature. Since 2018, he has been working with a preprint community to help develop standards and practices to promote the discoverability and reuse of preprints. So we will have basically two parts of the talk. The one is going to be on literature, on papers, and the second is going to be on data. And I invite Matt to start his presentation.
Thank you, Thomas. Let me just share my screen.
And it seems that Matt is frozen. Is that possible?
Can you see me now?
No, here we go. Great.
Excellent.
Matt, can we hear you?
Am I muted? Can you hear me?
Yes, we can hear you fine, Matt. Go ahead and proceed.
Excellent, thank you. So, thank you, Thomas, for the introduction.
I don't hear Matt. I do not hear Matt. I don't know whether that problem is on my side.
Okay. I'm assuming that everyone can hear me, except possibly Thomas.
Yeah.
I also don't see.
Okay, that could be on your side, Thomas. Perhaps shut off your camera to save some of your bandwidth. Matt, we can hear you and we can see your slides, so go ahead and proceed when you're ready.
Okay, thank you. So as Thomas just said, I'm Matt Pearce. I'm the technical project lead on the EBI search side of the web production team at EMBL-EBI. And I'm going to be talking about data sharing with the COVID-19 data portal today. So the COVID-19 data portal is part of the European COVID-19 data platform. That's a collaborative effort between the European Commission, EMBL-EBI, Elixir Europe, various EU member states and research partners. It's a priority pilot to realize the objectives of the European Open Science Cloud, an environment for hosting and processing research data to support EU science. The European COVID-19 data platform, includes a federation of national data portals hosted in those nations. This includes Italy, Japan, Norway, Poland, Slovenia, Spain, Sweden, and Turkey as of early 2021. So the data platform consists of 3 connected components. The SARS-CoV-2 data hubs, which organize the flow of the SARS-CoV-2 outbreak sequence data and provide comprehensive open data sharing for the European and global research communities. The Federated European Genome-Phenome Archive, which provides secure control access sharing of sensitive patient and research subject data sets relating to COVID-19, and the COVID-19 data portal, which brings together and continuously updates relevant COVID-19 data sets and is the main focus of my talk today. So the COVID-19 data portal was created in April 2020. The team was initially seconded from teams across EMBL-EBI, but since then, members have been hired directly to the team. We've had nearly 5 million views since the launch, and you can see it at the URL given here, the covid19dataportal.org. The patient data comes into the platform through national health data infrastructures and public health laboratories. Viral data goes through the SARS-CoV-2 data hubs, while the patient data goes through the federated EGA side. The data is processed and curated through the European Strategy Forum on Research Infrastructures and is made available through the COVID-19 data portal. In the COVID-19 data portal itself, the data comes from across EMBL-EBI and other institutions. The data that you see is a mixture of subsets of other data sets where we're actually filtering queries to restrict the data to COVID-19 specific entries or specific data curated especially for the COVID-19 data portal. It's updated daily and accessed through the EBI Search API. The EBI Search is a Lucene-based full-text search engine, and it has a full-featured API for searching, which is used by the COVID-19 data portal and other sites across EMBL-EBI. So you can see we've had a large number of teams contribute data. I hope I didn't miss anybody when I was pulling the slide together. I'm obviously not going to go through everyone here, but this is a list of the contributing teams. So the COVID-19 data portal has over 2 million records across molecular platforms and literature. It provides access to data resources and tools, over 70 linked related resources, and provides a web portal, an API, and download functions. And you can see through the timeline here how the the data has grown since January 2020, when the SARS-CoV-2 genome was initially submitted, through the launch of the COVID-19 platform at the start of April last year, and through to February this year. So there are several categories of data searchable through the COVID-19 data portal. These range from viral sequences and host sequences through to imaging, literature, and those related resources that I mentioned with the previous slide. And you can see here, this is a snapshot from the web page of the categories that you can search across in the portal. The categories themselves are accessed via tabs. Each tab has its own search box to allow initial querying. By default, all results from the top category in the portal are shown. So if I click through to host sequences and then expressions, you'll see the example searches here change as I click through. The results display allows you to navigate through the data types that fall into the category. You can see here where this is the viral sequences category, and we're looking at the sequences data type. If I click through to raw reads, you'll see that the layout changes depending on the data that we're looking at. You can click on the accession or the title, depending on the data type, to go to the original entry on the on its original location on its EMBL-EBI site. I click here. You can see we go through to ENA, where this record lives. One of the most powerful features of the COVID-19 data portal is the cross-reference function. It allows navigation between different data sets and allows you to cross-reference entries between the current data set and other sets that it lives in. The cross-references might be maybe internal or external, so they may be outside of the COVID-19 portal, but within EBI search, or sometimes actually outside of EBI search entirely. To change the results display, you may have noticed, if I hop back a slide, you can't actually see the full data that's available in this table. So we provide a horizontal scroll bar at the top of the table here to allow you to scroll left and right. We also have the function through the edit table view to change the table columns that are on display. You can also change how the cross-references are displayed, either restricting them to just the internal cross-references or to show everything, and whether those cross-reference rows are expanded or collapsed by default. So data can be queried programmatically through the portal API. It's a RESTful API where you could specify a query, which fields you're looking for, page size and number, and you are limited to 1,000 items per request per page. There are endpoints per data type, and you can, as I say, you can query the portal that way. There are additional query options available through the EBI Search API if you want to do cross-reference searches and so on. The COVID Portal API is a subset of the functionality available through EBI Search. It's important that it should be easy to download the data. A number of the data types that we have feature a download link directly on the page, allowing data to be saved in various formats. You can see here for the sequences data, you can save in EMBL or FASTA format, or you can just save the list of IDs that you're looking at, or tab-separated metadata, which will be the accession, the ID, and other metadata depending on the data type that you're looking at. You can also download bulk data for several categories. There's an FTP site link in the header of the site. You can also use Aspera, which is a command line download client. And there's a bulk download tool currently in development, which is a standalone Java application allowing you to download the data directly from search. We've got a demo coming up for the bulk download tool as part of our technical track talk on Friday, the 30th of July. And so I'd encourage you to attend that if you're interested in that functionality. So the COVID-19 data portal is built on FAIR principles. The data and associated metadata should be findable. It should be easy to find both for humans and computers. It should be accessible. So once the data has been found, it should be clear how to access it. It should be interoperable. So the data usually integrates with other data, presented in our case most often as cross-references. And additionally, vocabulary should be common across the data sets to make this simpler. And finally, the license for the data allows it to be reused in conjunction with other data sets. So the data is reusable. We have an open letter on the site setting out our aims, that the COVID-19 data portal provides open data and promoting data sharing through the COVID-19 data portal. At the time this screenshot was taken, we had nearly 800 signatures. We encourage you to sign it yourself if you haven't, and if you want to join the scientists who support the data sharing principles of the COVID-19 portal. We also invite data submissions. The instructions vary depending on the data set that you wish to contribute to. For viral sequences, there is a drag and drop tool available. The drag and drop function is dependent on a suitability check. You sign up with your email address and supply some sample data, and if suitable, you'll be given an open ID, I believe, allowing you to drop your data in. In terms of additional resources, there have been a number of papers published on the COVID-19 data portal. This one is the most recent in nucleic acids research, published, I believe, in May 2021. And if you've got a copy of the presentation, you can see it at this DOI.org link attached. But thank you for listening. To inquire on how to collaborate on the European COVID-19 platform, we have email addresses, and the email address is on this slide. To also share your data, we have a contact form on the site for other inquiries, and we also have a couple of technology track talks coming up at the end of July related to the COVID-19 portal, including that demo of the download tool if you're interested. So thank you very much, and I'll now hand over to Michael, who's going to talk about literature.
Thanks very much, Matt. I'll just start sharing my screen. And if someone could give me a thumbs up that you can see, that would be great. audible? Yes, would be great.
Yeah.
Brilliant, thank you. So I'll be following on from Matt by just taking a small step away from the data itself and towards the literature in which that data is both reported and reused. I'll specifically be talking about a project within the literature services team at the EBI to improve the accessibility of COVID-19 preprints. So a quick introduction to Europe PMC, which is one of the main resources developed in the literature services team at the EBI. It's a database of biomedical and life science research and a repository of choice for a number of international research funders. We are a member of PubMed Central International, which means that PubMed Central shares with us their full text articles. And these full text articles are captured in a standardized and highly structured document format known as JATS XML. The majority of these XML files and the associated figures and supplementary files are supplied by publishers, but there's a smaller but appreciable set that are deposited directly by authors via a submission system that we run called Europe PMC+. In addition to the full text content from PubMed Central, we also include journal abstracts from PubMed, and since 2018, we've also been including preprint abstracts alongside the peer-reviewed literature to improve their discoverability. For anyone that's not familiar with preprints, as a quick introduction, they are academic manuscripts that are uploaded to a public server before they undergo peer review in a journal. The key aspect relevant to this talk is that they allow scientific results to be shared far more rapidly than traditional journal publication, because there isn't a requirement for a formal peer review process, which can of course take many months. We currently index over 300,000 preprints, along with relevant information. For example, we include links from preprints to final journal publications. Preprints are included in our citation network, so you can see which preprints are being cited in the journal literature. We attach ORCIDs to preprints. ORCIDs are unique identifiers for authors, and we allow the ability for authors to claim preprints to their ORCID record. We also provide links to the data that's referenced in preprints. We link to peer review reports and other commentary. And finally, we link to lay summaries. On the slightly more technical side, preprints often have a version history, which is not something you typically see in journals, which we maintain in Europe PMC. You can see on the left a preprint with 4 versions and the ability to navigate between those. You can also see in the orange banner a bi-directional link from the preprint to the final journal publication. But in our search results on the right, we only include the latest version of a preprint to avoid unnecessary duplication within the search results. In the context of the pandemic we still find ourselves in, preprints have played a very significant role in the rapid dissemination of COVID-19 research findings and the associated underlying data. While the number of preprints is small in the life sciences in comparison to the peer-reviewed literature, by the end of April 2020, a few months after the beginning of the pandemic, preprints accounted for about 1/3 of all COVID-19 literature indexed in Europe PMC. The graph you can see on the screen looks at COVID-19 preprint publication over time, and there are 2 things to draw your attention to. Firstly, that we see a significant number of COVID-19 preprints being published from late March, which, considering the timescale of the pandemic, is quite a short lag time. Secondly, this is quite a colorful chart, which reflects that we have preprints being published in a very wide range of servers. It's important to note that after these preprints are uploaded to these public servers, they're typically only available to the user as a PDF file. Sometimes there might be an HTML conversion. Furthermore, each preprint server has its own interface and its own search syntax, So for anyone who wants to do any large-scale analysis of the COVID-19 preprint literature, they're going to face a problem because they will need to search multiple servers, download lots and lots of PDFs, and then do perhaps some text extraction of what are generally very poorly structured PDF files. Early on in the pandemic, an initiative originating from the White House Office of Science and Technology led to a number of publishers making the full text of COVID-19 related journal articles free and publicly available via PubMed Central. So as a complementary effort, in the spring of last year, we proposed a project to convert COVID-19 preprints from a range of servers into this structured JATS XML format. And happily, this work received grant funding from the Wellcome Trust, from the Medical Research Council here in the UK, and the Swiss National Science Foundation. And it was further endorsed by the chief scientist of the World Health Organization. The idea here was to open up the full text of preprints in a very structured, machine-readable format for reuse by anyone interested. As a very brief overview to the approach here, this involves retrieval of the manuscript PDF and associated files for a COVID-19 preprint. In this case, it's from a server called medRxiv. This is done using some Python scripts. An external vendor we contract to actually do the conversion of these PDF files to this structured XML. These XML files are reviewed by the author within a submission system that we run called EuropePMC+. There is then an indexing process at EuropePMC, which makes the full text available to read on the website. And finally, we make these files available to download either via the public API or via FTP. I'll just briefly note here that preprints are uploaded to servers under a variety of licenses, which affects what can be done downstream. We don't make the preprint full text readable or downloadable where either there's no Creative Commons license, which allows us to do so, or the author has not given us permission to do so. So once these preprint XMLs from various repositories are indexed in EuropePMC, there's now a single source that a user has to go to for a comprehensive search of the COVID-19 preprint literature. EuropePMC has a very configurable search syntax, which at its simplest can be used to find preprints with mention of a keyword or combination of keywords within the full text. In the example on the left, I'm searching for COVID-19 preprints which mention the spike protein anywhere in the full text, and I'm receiving just under 2,800 results. I could further refine my search with additional keywords, or perhaps look within a certain range of publication date. And there are also some options for sorting this result list, for instance, by relevance to my search query or the number of times a preprint has been cited. Alternatively, on the right-hand side is our advanced search feature, which lets you run much more complex queries. Perhaps, for example, looking for COVID-19 preprints with keywords in specific sections of the full text, the data availability statement, for example, or to find COVID-19 preprints with data citations to specific resources, such as the Protein Data Bank, as well as other fields like funding, presence of peer review reports, license type, and so on. One of the key benefits for us and for others of using this JATS XML format is that within our team, we already have many processes that run on our full text journal content from PubMed Central. We run these same text mining pipelines on our COVID-19 preprint XML and extract various biological terms, entities, identifiers, and relationships, which are displayed alongside the preprint on our website through an application called SciLite, which you can see on the slide. We have a huge number of such annotations across all our content. Further text mining groups from across the world can also freely download these preprint XMLs, license permitting, and turn their own software onto these COVID-19 preprints with minimal extra effort in order to extract the information that's valuable for their particular audience. For EuropePMC funders, we also extract grant information from the full text. Funding metadata can be fairly patchy even in the journal world, but it's pretty much non-existent in preprints. So these text mining processes will only capture a fraction of the funding because it requires the author to have written it into their preprint, but it's better than none at all. Although our focus has been on generating these full text XMLs, with a very convenient bulk download, it's very easy to do some large-scale analysis on the COVID-19 preprint literature. Just by way of example, on the left are two pie charts comparing the proportion of COVID-19 preprints that we found links to data records in comparison to full-text journal articles on the bottom. And on the right-hand side is an analysis of the licensing breakdown on a repository level. For example, we see that CC BY NC ND is the most common open access license among the COVID-19 literature. In terms of reuse of this data, we've seen already. Firstly, there's an organization called Prelights who publish written highlights of preprints. They've created a COVID-19 timeline that combines important events that have happened throughout the pandemic alongside the dates at which important preprints have been published. ASAP Bio, which is a non-profit preprint advocacy group within the life sciences, they're directing readers to some of the data you've seen earlier in these slides. And finally, a text mining group at the Swiss Institute of Bioinformatics are using a coronavirus ontology to generate annotations that relate to COVID-19 concepts. And they're turning their software on these COVID-19 full-text preprints. We've converted around 28,500 COVID-19 preprints so far, and we've been adding service progressively over the last year. The chart you can see reflects all of the COVID-19 preprints available in Europe PMC by month. The yellow are ones that are available to read. The blue are cases where the full text is searchable, but not available to read. And in the gray, we have just abstract-only content, which is either preprints that are currently in processing or for preprint servers that we don't yet include as part of this project. So this is still very much a live project. We're converting around 600 preprints a week. There's been a slight slowdown recently as we added a well-known repository called Archive, which contains a lot of modeling studies and equations that are complex to capture. In terms of our future plans, we hope to continue converting all COVID-19 preprints into this structured format, at the very least while the WHO still considers that we are in an active pandemic. Of course, we don't expect COVID-19 literature to suddenly disappear after that, so this will largely be down to future funding. We'd like to add more preprint servers, and finally, we'd like to work closely with the preprint community on standards, especially around the withdrawal and removal of preprints. This is analogous to retractions in the journal literature, as well as better surfacing peer review and other commentary around preprints, particularly relevant for COVID-19 preprints. With that, I'll close, and I'd just like to thank the organizers for the opportunity to talk today, and to acknowledge the 33 funders of Europe PMC, in particular the 3 mentioned earlier that funded this work. I'll now stop sharing my slides and we'll go to a discussion.
Thanks a lot for both of your talks. I have 2 problems now. One problem is that I had a software issue through a large part of Matt Pearce's talk, which precluded me from hearing his voice and seeing his slides. So I felt a little bit like Helen Keller, which is dramatic, which also makes it difficult for me to ask questions. I will have questions nevertheless, but you may have answered all of them in your talk. Issue is that we have a Q&A feature and people have been active in the Q&A feature, but you've also had little angels answering all these questions online. So we may be in a lack of unanswered questions, but we'll see. We're interminably flexible. Do you hear me? Do you still hear me? Okay. My window just told me that my connection is interrupted.
That's okay. So yes, we can still hear you, and I think Maria has been the little angel answering them in the background. But if we have the time and we are recording, so what I would recommend we do is go ahead and we can reiterate those online. So if you'll ask the questions, Thomas, between Matt, Michael, and Maria, we'll go ahead and re-answer those so that it's part of the recording.
Okay. All right. So I'll go through the questions. There was a question about the license for data in the data portal, right?
Yeah. Is licensed through Creative Commons. There's a terms of use link on the main EBI front page. But the data for the most part is Creative Commons licensed.
Okay, thank you. I want to add a question, a personal question, a question by myself on this in a more general context. We see in the COVID-19 area, we see a fragmentation of the datascape. There is GISAID, and there is GenBank, and there is your data portal, and I'm not quite clear about what exactly the relationship is. So GISAID has more data than you have. I just looked at the portal. There are 750,000 sequences in your portal. There are more than 1 million GISAID. Is there any overlap between those? Is there double submission? Do we have an overview? Is there a difference between what you have in your portal and what GenBank has in its portal? Can you address that?
I'm sure there is an overlap between the two.
You're not using GISAID data, right? You don't extract data from GISAID for your portal.
No, we don't. Our data, our sequences data comes through the ENA.
Right.
So yeah, I'm not actually sure what the how much overlap there is between the data.
So do you import data from anywhere, or do you rely on proactive submissions by people who submit these sequences?
So, we're relying on submissions basically through the SARS-CoV portals.
Okay.
And the data that comes in is curated by other teams in EMBL-EBI and supplied to us. All
right, thank you. That is impressive. If you don't import, if you just rely on submissions, then you have so many sequences. There must be a lot of double submissions there, which is good. We do
have a document on the outbreak data, and yeah, my colleague who's in the chat has clarified that the INSDC data is actually a combination of the GenBank, ENA, and DDBJ data.
I see. And your data are ENA?
Our data is ENA, yeah.
Your data is ENA. Okay, good. Thank you. I'm looking for other questions. There was a question on EuroPMC, whether it provides APIs to access both peer-reviewed journal articles from PubMed, PMC, Agricola, and CBA, and preprints as well as other documents. But that was answered in the affirmative.
They're not currently available online. Okay. I'd be happy to send you a PDF of them.
That would be nice.
After the meeting's finished.
Great.
So I'm gonna jump in really quick too. Something that if you would like to do, if you could both send Seth the links to your slide, he can add a frame at the very end of this where we can put the links to those for you on the recording.
Okay. I have another question for Michael, which may also be something you said, but I didn't understand. So you said that you have these preprints online, and you have the version histories of the preprints online. So what happens when a preprint gets published? Is that version history continuous through publication, or do you enter a different world then, the published world? Or does ever, if you have version 17 of a preprint, does version 1 ever go away, or do you have a complete track of everything?
Yeah, that's a really good question. So from our side as an archive, we feel it's important that there is a complete version record. That is, because there might be a version 17, it's very important to know, well, maybe when was version 1 published? That
Yes.
is important because it is still part of the same record.
Right.
So the way this works is version 1 of a preprint is published and it gets added to Europe PMC. If any subsequent versions come along at a later date, they get added to essentially a stack of versions that are navigable between. And when a final journal publication arises from the preprint, not all preprints result in a final journal publication, that will come through PubMed or PubMed Central. And what we do is we provide essentially a clickable link on the website that will take you from the preprint version, whichever version it happens to be, to the journal and to the journal article record within Europe PMC. So it looks very much the same. There may be full text for the journal, that article there may not be, but they are considered distinct for reasons such as, is a citation of the preprint also a citation of the journal article? It seems perhaps not. We find most people will, when they find a preprint is published in a journal, they will switch their citations to citing the journal article instead of the preprint, which makes sense. Furthermore, is each preprint cited, if a version specific citation is made, how do we count the citations? Do we aggregate all version citations in one? So what we do is we try and keep the version as transparent and complete as possible and allow the user easy access to navigate between the versions. So that's the way we do it, rather than collapsing everything into one single record.
All right. Thank you, Michael. I assume you finished your answer. I lost you again. So
hopefully the recording
need to press Control F5 here in order to stay alive. Thank you. Just I want to add another question, since I don't think there is another question in the chat. No, there is not. So with living documents starting to appear on the scene, article trajectories can be quite complex. I mean, they don't need to be a path. So do you expect in the time to come, I assume that you rely on a path as a trajectory for an article and at a single entry point for a journal article. So just, thinking about the worst possible, this could all become much more complex. So what are your ideas on this? Am I too pessimistic here? And are we going to stick with paths, or how are you going to prepare for this complicated world?
Yeah, you're absolutely right. The closest thing we see already existing and having existed for a few years are so-called open research platforms. So if you're familiar with the publisher F1000, their model of journal publication is quite unique, where the initial submission is to a journal, peer review is open, and the revisions of the manuscript are published as subsequent versions. And at some point, one of those versions becomes published. It gets peer review approved. And so even now, the workflow is not quite as straightforward as it used to be. With preprints having versions, we think we've got quite a robust system for handling versions and changes between them. When manuscripts start to move to living documents, perhaps, including code within the document, interactive graphics, perhaps, revision changes happening live, annotations on things. Yeah, this sort of static model of publishing becomes quite problematic for an archive like Europe PMC, which has been built up on a sort of model of there being a PubMed abstract. We are quite adaptable. We're lucky that we're quite a flexible organization. We're supported here at the EBI. And we try and be innovative. And so I think we're in a good place to adapt to those changes. We haven't seen this happen yet. There hasn't been a need to host something like a real living manuscript, but it is probably around the corner.
Okay, thank you. I'm always thinking very far ahead, so some of these things might never come. And I don't think we have any other questions. I want to ask you a last question, both of you actually. So this, in some sense, this has been a webinar in two parts. Matt talked about the data, you talked about the papers, and my question is there a link between the two of you? Are you ever in the need of talking to each other? Is there a link to the portal from Europe PMC or the other way around, something that requires you to interact and interface?
The literature section on the COVID-19 data portal is actually Europe PMC data. So we certainly interact in the background, although we haven't had a great deal of interaction before preparing for this talk.
Just give a brief answer from the Europe PMC side. So one of the things we really try and put as much effort into as possible in Europe PMC is linking up the literature with the data. And so we are linking the literature to lots of databases in the EBI, a lot of which form part of this COVID data portal, and of course other databases.
All right, thank you. I don't think, let me see, is there another question? No, they're just answers. Very good. Okay, I think I will close the webinar here. I want to thank you very much, both of you. You have great resources that you give to the public, and we hope that with the recorded version of this, we will add our little bit of helping you to even increase your visibility more. We are thankful for your work. We're thankful for your talks, and I'm thankful for the listeners, both the listeners live now and the listeners in the future. And I wish us all a good rest of the day. Thank you.
Thank you very much.
Thank you.